Hello and welcome to the Pharma Training. Today we will discuss about the basic of the analytical method validation. In this topic, we will discuss the basic, what is the basic requirement in a analytical method validation. So in this session, we will discuss what is the validation, what is the verification, validation parameter, and the validation study. So first, what is the validation? So first of all, in a quality control department, we are using the many method like the SA by HPLC, RS by HPLC, dissolution by HPLC, UV method, TLC method, water content, identification method. All methods should be validated before the use. So method validation is the process of the determination of the method that method is fit for the intended use and to provide the framework of the ensuring the accuracy, reliability, and consistency of the analytical method used in a drug development and quality control department. So, method before we use the method, we have to ensure that the method should be accurate, reliable, and consistent. If the method is not consistent, then we can get the uneven result during our routine analysis. So, before use any method should be validated. And method validation is usually applied to an in-house method developed by the laboratory. Whatever method we develop the in-house, those methods should be validated. What is the verification? The method verification is the process to confirming the method that the method is suitable for the laboratory use. The method verification is applied to the compiled method or previously validated method when it is being used in a particular laboratory for the first time. The method it is already available in a pharmacopoeia, Indian pharmacopoeia, British pharmacopoeia, USP, EP, JP. It is already available in a pharmacopoeia. Then method validation is not required. For that, we have to verify the method. The method is suitable for the use in our laboratory. And another thing, if we use the already validated method by the any other user. Like in a raw material, we are using the vendor method for the residual solvent or the any in-house method or product for the SARS. So for that, we have to verify the method in our laboratory because of the method is already validated by the vendor. So in our laboratory, we have to just verify the method. What is the parameter of the method validation? So before start the any method validation, we have to start with the specificity study. Whenever we start the method validation, so first we have to start with the specificity study. Then LOD and LOQ determination. Then after the determination, we have to confirm the LOD and LOQ. Then perform the linearity and range, accuracy, precision, solution stability, robustness, and filter study. So about the myself, I am the Bhavik Jani, 20 plus year experience in a pharmaceutical formulation, work in a quality control, quality assurance department, work as a QAIT, QF, QC, work in a major organization like the Sun Pharma, IPCA laboratory, Unique laboratory, Umedica, and the Corona remedies. And if you require the uh, presentation of the assay, dissolution, reactive substance, or cleaning procedure, how to prepare the protocol and the report, how to calculate the concentration, you can contact me or WhatsApp me on this number. So first we will discuss about the specificity. The specificity is the ability to access the unequally the analyte in a presence of the component which may be expected to be present. Typically, this might be including impurity, degradation product, or metric product. In a simple language, if we discuss the specificity study, so when we run the standard sample, blank, placebo, dilute, so many peaks are there. In a blank, one or two peaks are there. Placebo, one, two, three or peak are there. Impurity peak are there. So The purpose of the specificity study is each and every peak should be separated. The absence of the relevant interference with the product, the buffer or appropriate metrics, and the between the individual peak of interest means all peak, blank, placebo, impurity, standard, sample, all peaks should be separated. Peaks should not be merged with each other. Then we have to spike the impurity in our test sample. And after the spiking, all the impurity peaks shall be separated with each other. Demonstration of the stability indicator property through appropriate post-degradation sample if necessary. If this method we are going to use in our stability study, then methods should be validated by the post-degradation study. We have to ensure that by post-degradation study, any additional peak are not generated. If are the generated, which is not merged with the impurity or any other peak. So how to prepare the sequence in a HPLC? So first we have to inject the mobile phase or blank or dilute, then placebo of the older strength, then excipient if required, then system suitability injection available in a method, standard solution 5 or 6 as per our SOP, identification of the each impurity. If the method is available, the four impurity A, B, C, D, so you have to prepare the individual identification solution for the A, B, C, D and inject those solution. Then the sample preparation and sample preparation with the spike. You have to spike the impurity in a sample preparation. And what is the acceptance criteria? The no interference should be observed in retention time of the active ingredient impurity degradant peak from the dilute and placebo. Whatever peak observed in a dilute and placebo, it should not be interfere with the impurity or main peak or standard peak. Impurity peak should be well separated and peak purity should be more than 1.0. Your peak should be a pure. Specificity should be established from the first degradation study. First degradation study is required if the method is used for the stability indicator method. Now the first degradation to be performed as per the requirement. First degradation study is performed as per our requirement and as per our product experience. So in a first degradation study, we have to perform the acid degradation, alkali degradation, peroxide degradation, UV degradation, thermal degradation, water hydrolysis, acid, alkaline, and water, and photolytic study. This type of the degradation we have to perform during our methylation study. In a first degradation, we have to inject the extract sample and the treated sample. Treated sample means degraded sample and calculate the difference of the both sample. Calculate the peak purity of the main peak and the impurity peak and calculate the mass balance. Now LOD and LOQ. What is the LOD and what is the LOQ? The LOD means limit of the detection. The detection of the limit of individual analytical procedure is the lowest amount of the analyte in a sample, which can be detected but not necessary to quantify with the exact value. Means in your homogram, the lowest amount of the analyte means smallest peak, which detected, but you cannot the quantify with the accurate. Means if you inject the 5 injection or 6 injection of the same sample, then it is not necessary to you can get the same area for all the 5 or 6 injection. So lowest amount of the analyte which can be detected but not necessary to quantify means peak will be detected, but you cannot quantify with the accuracy and precision. Then limit of the quantitation, LOQ. The quantitation limit is the lowest amount of the analyte in a sample which can be quantitatively determined with the suitable precision and accuracy. Means in your sample chromogram, the small peak are observed with you can quantify with accuracy and the precision. Means if you run the 5 to 6 sample from the same vial, so you can get the same area of the those the smallest peak. You can get the same area for the all the smallest peak, and you can calculate the percentage RSD for that. The quantitation limit is a parameter used for the quantitative assay for the low level of the compound in a sample metric, and particularly is used for the determination of the impurity and degradation product. Means LOQ is required for the residual solvent and related substance method. There is a three types of the method to determine the LOD and LOQ. First, based on the visual evaluation. Second is based on the signal to noise ratio. And third, based on the standard deviation and the response and the slope. The first, based on the visual evaluation. Visual evaluation may be used for the non-instrumental method, but may be used with the instrumental method. The determination by the analysis of the sample is known the concentration of analyte, and by establish the minimum level at which the analyte can be reliably detected and quantified. Visual evaluation means like the limit test where there is no quantification are there. So where we have to just check the color of the standard of the sample. And in a TLC, we just observe the spot. So by this method, we can evaluate the LOD and LOQ. And based on the signal to noise, this approach can be applied to the analytical procedure which exhibit the baseline noise. Whenever the analytical method where the baseline noise is in a higher side, in this case, you have to determine the LOD and LOQ by the signal to noise method, where the baseline noise is the higher side. Determination of signal to noise ratio is performed by the comparing the measure the signal from the sample with the known low concentration of the analyte with those the blank sample, and establishing the minimum concentration at which the analyte can be reliably detected. And you have to check the signal to noise ratio. The 3GM1 is generally considered the acceptable for the estimating the LOD, and for the LOQ, it should be at least 10GM1. The signal to noise ratio, how to calculate the signal to noise ratio. This image I have taken from the USP, where you have to calculate the signal to 2h divided by small h. This image is explaining the how to calculate the signal to noise ratio. And third is based on the standard deviation of the response and the slope. The detection limit may be expressed as a detection 3.3 sigma divided by the slope, and the quantitation limit is 10 sigma divided by the slope. So now we will understand how to determine the slope and the sigma standard deviation. The sigma is the standard deviation of the residual response, and the S is the slope of the calibration curve. For the example, if you prepare the five solution, the concentration is the 5, 10, 15, 20, and 25. And the area is the for the 5 ppm, the area is the 4800. For the 10 ppm, 11250. For 15 ppm, 15421. 20 ppm, 20512. And 25 ppm, 26854. And when we draw the linearity graph, so our slope is 1067.4 and the intercept is minus 243.6 and the R square is 0.995. So how to calculate the residual response? So calculation of the residual response is the response minus slope into concentration minus intercept. For example, our response is the 4800 minus our slope is 1067.4 into 5. Concentration is the 5 minus our the intercept minus 243.6 and the result is the minus 780.6 residual response. So by that we have calculated the residual response for the all the concentration and calculate the standard deviation. Standard deviation is the 591.3960. Now we are going to determine the LOD and LOQ. So LOD 3.3 into standard deviation divided by the slope. The formula is that 3.3 sigma divided by slope for the LOD value and for the LOQ value 10 sigma divided by the slope. So 3.3 5 into 591.3960 is the standard deviation and divided by slope. So your LOD value is 1.82 ppm and your LOQ value is 5.5 ppm. By this method, you can determine the LOD and LOQ value. Hmm. So how to prepare the sequence for the LOD and LOQ determinants? For that, you have to inject the blank and placebo first, then the system suitability available in your the MOA, then the standard injection 5 or 6 as per our method of analysis, then standard or impurity concentration 1, 2, 3, 4, 5, 6, 7, 8, 9, and inject the bracketing as per your method of analysis. Now LOD and LOQ conformation. For the LOD and LOQ, after determination of the LOD and LOQ determination, you have to confirm that. So how to prepare the sequence? First, you have to inject the blank or mobile phase, system stability, standard as per the MOA 5 or 6. Then LOD level, whatever level you determine, you have to inject the 6 times, same concentration. And the LOQ level, whatever the LOQ level you determine, you have to inject the 6 times. So in this case, we have determined the LOD value is the 1.8 ppm. So you have to prepare the 1.8 ppm solution and inject the same solution in the 6 times. And LOQ, we have determined the 5.5 ppm. Then you have to prepare the solution of the 5.5 ppm and inject the 6 times. And percentage RSD of replicate injections should be as per the acceptance criteria. Whatever criteria we define in our MOA or the protocol, it should be within the limit. Now the linearity. Linearity is the ability of the test to produce the result that are directly proportional to the concentration of the analyte in a sample. The range of the analytical procedure is the interval between the lowest and the highest result in a with the analytical procedure has a suitable level for precision, accuracy, and response. So how to prepare the solution? So dilute the analyte of the interest over the accepted procedure range at least 5 point up to the LOQ level for the impurity. For the linearity, you have to prepare the minimum 5 level from the LOQ to 150 percentage. Like the for the related substance, prepare the solution for LOQ level to 150 percentage, at least the 5 point concentration. In this case, we determine the LOQ is 5.5 ppm. So you have to prepare the 5.5 ppm solution. Then the might be a 20 percentage, 50 percentage, 100 percentage. Then the 150 per percentage, you can prepare the at least 5 point concentration for LOQ to 150 percentage. For the assay, prepare the solution from the 50 percentage to 150 per percentage, at least the 5 concentration. For the assay, you have to prepare the 50 to 150 percentage. And for the dissolution, if the immediate release, then prepare the solution from the S3 minus 20 to 150 percentage, at least the 5 point concentration. For the immediate release, S3 means if your the limit is 70 Q, then your S3 will be a 45 percentage. So 45 minus your 20 means linearity should be performed for the 25 percentage to 150 percentage. For the immediate release, S3 minus 20. And for the sustained release, you have to prepare the solution for the 1 percentage to 150 percentage, at least the 5 point. Because of the sustained release tablet, limit might be not more than 20 per percentage for the first hour. Means your result, if you get the result the 1 percentage, it will be a pass. So you have to minimum cover the 1 percentage to 150 percentage for the linearity and the accuracy. For example, LOQ level and 20, 50, 75, 100 and 150 percentage. You have to prepare this type of the solution and the inject. The sequence will be a blank or mole phase, system suitability standard as per the MOA 526. Then uh, standard or impurity concentration, LOQ level, 20 percentage, 50 percentage, 75 percentage, 100 percentage, 150%. Then you have to inject the bracketing standard as per your procedure. The correlation coefficient for the linearity should be not less than 0.995. Now the precision. The precision of the analytical procedure is expressed the closeness of the agreement between the series of the measurement obtained from the multiple sampling of the same homogeneous sample under the prescribed condition. So there are three types of the precision. One, the repeatability means system precision or method precision, intermediate precision, and reproducibility. So now what is the repeatability? In a repeatability, there are the system precision. Inject the standard or system suitability, and it should be meet the acceptance criteria. In your method, if the system suitability criteria is given, like resolution, theoretic plate, or scaling factor, or percentage RSD of the standard, so you have to inject those the standard and system suitability, and it should be meet the X per your MOA acceptance criteria. And the method of precision, reparability express the precision under the same operating condition over the short interval time. Reparability also term the intra-assay precision. So in a method of precision, how to prepare the solution? Replicate measurement with the 3 times and the 3 level across the reportable range or 6 times at the 100% level, considering the peak of interest. Replicate measurement with the 3 times and the 3 level means you can prepare the 3 level means 50 percentage, 100 percentage, and 150 percentage 3 times. But three sample of the 50 percentage, three sample of the 100 percentage, and three sample of the 150 percentage, or 6 times at the 100 percentage means 6 times at the 100 percentage you can also prepare for that. And method precision should be performed with the RRF and external standard if the method is for the external standard. for the related substance test. If your calculation is from the RRF method, then you have to perform the method precision by the RRF and the external standard in a both method. The acceptance criteria, percentage RSD of the 6 preparation or 9 preparation should be as per the protocol. Percentage RSD of the 6 preparation or 9 preparation should be as per the protocol. And recovery difference between RRF and external standard should be plus minus 20% of the mean. So in method precision, you have to prepare the 3 times or 3 level sample or 6 times at the 100% level. In intermediate precision, study to be performed same as a repeatability, but with the different days, different instrument, different column, and the different analysts within the laboratory. You have to perform the study with the same as per the repeatability, but they should be different, instruments should be different, columns should be different, analysts should be different within the laboratory. And samples preparation, replicate measurement with the 6 times at 100% level, considering the peak of interest. Then acceptance criteria, the percentage RSD of intermediate precision, method precision should be as per the protocol. You have to calculate the combined percentage RSD of the intermediate and the method of precision. Now the reproducibility. Reproducibility expresses the precision between the laboratory. Inter-laboratory study usually applies for the standardization of the methodology. If you have a two lab A and lab B, if you develop the method in a lab A and method validated in a lab A, then whenever you transfer the method in a lab B, this called a reproducibility. Means the different laboratory. The solution prepared the replicate the measurement with the 6 times at the 100% level, consider of the interested peak. Reproducibility is the one type of the method transfer. The percentage RSD of reproducibility and method precision should be as per protocol. H12C sequence is the blank or mobile phase, then system suitability, standard, sample 1, 2, 3, 4, 5, 6, and the bracketing as per your MOA or the procedure. Now the accuracy. The accuracy of the analytical procedure express the closeness of the agreement between the value which is accepted, either the conventional true value or accepted reference value, and the value of the set measure. Solution of preparation. The spike a standard or impurity in a placebo with different concentration level. For example, prepare the solution 50%, 100%, and 150%, and LOQ concentration level. So in accuracy, you have to prepare the four type of the solution. LOQ level to 150% level, like the LOQ level concentration 50% and 100%. And the same solution should be spiked in a sample. The same concentration should be spiked in a sample, and you have to calculate the recovery. The concentration to be considered for the 50% of the minimum limit and 150% of the maximum limit. The minimum limit means in your finished product might be a impurity limit is a 0.1 of percentage. So you have to calculate the 50% of the 0.1 percentage, and your stability specification limit might be a 0.2 percentage. Then you have to consider the 150% of the 0.2 percentage. That's why I mentioned that the 50% of the minimum limit and 150% of the maximum limit. And accuracy at the LOQ level should be performed for the date, and 3 sets for the each level to be prepared. And percentage recovery and percentage RSD, each level should be mentioned in a protocol. Sequence should be there, the blank and mobile phase, system suitability, standard solution as per the MOA, then the LOQ level 3 set, 50% 3 set, 100% 3 set, and 150% 3 set. Solution stability. Solution stability to be determined as per the method of analysis at the 100% level. And for the solution stability, you have to consider the mobile phase stability, diluent stability, system stability, standard and sample should be considered for the solution stability. Solution stability to be performed for the given temperature in a MOA, desktop study, stability at the desktop, and the instrumental study. You have to perform the both study while you have to fill the vial and kept in a HPLC rack, and you have to perform the solution stability of the desktop solution also. You have to fill the solution from the desktop every time and the inject. The sequence should be blank, mobile phase, system suitability, standard solution, sample solution. Then after 6 hour or 9 hour, 12 hour, you can inject this. The next is the filter study. For the filter study, you have to prepare the standard and sample as per the MOA. Then different type of the filter to be used for this study. Discard volume study to be performed before finalize the filter. You have to finalize the discard the volume, and one sample solution to be prepared and use the different type of the filter and discard the volume. Like the first, you have to first the sample the solution with the centrifuge. Then you can use the filter one, filter two, filter three, and the calculate the difference between the each filter. Then you can finalize your the filter. Robustness. The robustness of the analytical procedure is a measure of its capacity to meet the accepted performance criteria during your normal use. Robustness is tested by the deliberatory variation in a analytical procedure parameter. Following variation, but not to be limited: flow rate plus minus 2 ml, wavelength plus minus 2 nanometer, temperature, mobile phase ratio. You can variation in a pH plus minus 2 variation in a pH. And another thing also you can uh, perform the robustness by the sonication. If your method is given the 15 minute, then you can use the 12 minute or the 17 minute. So you can experiment as per your experience and the product behavior. Thank you all. If you have any query, you can contact me on the WhatsApp. You given on this number. Okay, thank you and thank you so much.